Very good morning, students. In this particular talk, we'll be dealing with the aspect of the evolution of the heart in the case of the reptiles. Now, in the previous sections of this particular topic, we had talked about the heart of the amphibians, in which we have seen that the heart in that particular case was a three-chambered heart. Now here also in reptiles we have got a typical three-chambered heart, but there will be a kind of a partial septum present also in the reptiles. So we will be seeing the case that how these reptiles they have modified the heart according to their needs and the environmental conditions. So these reptiles they are terrestrial in nature. So if we see the variety of the sizes in the body of the reptiles, they are quite big in size. So reptiles they show a diverse, you can say, modifications in the body sizes. So we'll be seeing that how they cope up with the modification in the heart with the environmental conditions in which they live. Now students, if I start with the reptilian heart, so basically this heart, it is present in the reptiles, and reptiles are the first forms to be truly terrestrial. Now this is what we have earlier discussed, that these are the truly terrestrial animals that are present on land. Now there are some exceptions like turtles and crocodiles to this particular habitat, but still all the reptiles they are terrestrial in their environmental conditions. So they have got the lungs as the primary source of the oxygenation. So as they are terrestrial, so they have got the lungs for the respiration. Now, if we go further, so there is an effective pulmonary respiration in case of reptiles. So it is quite effective because they have got a well-developed respiratory system. Now, as we have previously discussed, there is a huge size variation in case of reptiles. So they can be quite small and they can be as big as like crocs. They can be 5 or 10 meters long. So they are quite huge in size. So there is a large size variation. So due to this size variation, so we have got different variations in the heart also. So the size of the heart is typically larger in the reptiles as compared to the previous animals. So you will see that the heart, it has got various modifications as compared to the amphibians and the lower animals. Now if I go further, there can be huge sinus venosus in some of the turtles. So these are some of the variations which are present in reptiles, and many reptiles they have a reduced sinus venosus. So this is a very important point to note, that if they have got a reduced sinus venosus, the valves near to the junction of the veins and the right atrium, they are representing the remnants of this sinus venosus. So students, this is a very important point. So before going into this point, I must give you some idea which we have discussed in the previous lectures, that the sinus venosus was receiving the blood from the body. It was a deoxygenated blood and it had a connection with the right auricle. Now here, when the sinus venosus is you can say reduced in size, so it is now represented as the valves which are present near the right atrium. Now we don't have a sinus venosus, but we have the valves, and these valves, they are the remnants of the sinus venosus, which are present near to the right auricle. So this is an important point, and you should note it in the copy. Okay, if we go further in reptiles, what we have is now we are to the typical reptilian heart. Now, if I go further, it is a typical three-chambered heart, and more systematically speaking, it is 3.5 chambered. So, as we have seen in the case of amphibians, that can be considered as a 2.5 chambered heart, but we are considering as like a three-chambered heart. But this is also here, also in reptilians, we have got a 3.5 chambered heart. Now, this particular diagram is showing you that how it is 3 chambered or 3.5 chambered. Why I'm saying that it's 3.5 chambered heart. Now here we have got two auricles. These are the two atria, and we have got a single ventricle, but it is with the incomplete intraventricular septum. So I have written IV septum over here. So we have got a partially divided ventricle and two atria. That's why it is 3.5 chambered heart. But in the books you may find it as a 3 chambered heart in the case of reptiles. Now here we have the conus arteriosus, and it is also having a particular demarcation in between to keep the two separate streams, which will be going into the aorta to be distributed. And we have a pulmonary artery which is going for the lungs. Now students, if I go further, the conus arteriosus, it is also reduced to some extent. Now the ventral aorta, it is dividing into three main trunks directly. So as the conus arteriosus is reduced in this case, so the ventral aorta will be dividing into three main trunks. Now first trunk is the pulmonary arch. It will be going to the lungs. Second is the left systemic. So it will be going to the systemic, refers to the organs. So that will be going to the body organs. And we have a right systemic arch also. So we got two systemics and one pulmonary arch in the case of reptiles. Now both the right and the left systemic aorta, they cross each other. This is a very important point. They are crossing each other. So if left systemic is going like onto this side and right systemic is going into this side, so they are crossing each other at a particular point, and this point is very important in this particular heart. So at this particular crossing, what we have, it is called as the foramen of Panizza. Now this is the crossing, or you can say this is the joining of the two systemic arches. So this is called as the foramen of Panizza. So I'll be taking this particular pore later on. Now students, this particular diagram is giving you a recall about what we have undertaken in the previous lectures. Now if we go for an amphibian, we have got a 3-chambered heart. If we go for a turtle or a reptile, we have got almost 4-chambered heart, as we have said that it is a 3.5 chambered heart. Now if we go for a human or a mammal or a bird, so it is a typical 4-chambered heart. Now here this is a picture of a human with the crocodile. So he's a very famous crocodile man who I think you must have seen in him in Discovery Channel. So why I'm showing you this particular picture is that this human heart has got four chambers. We all know this, that we have four chambers. Now, how many chambers are present in the heart of a crocodile? Now, this is a question for you. In the previous slide, we have seen that it is three chambered or 3.5 chambered. So, my question is that how many chambers are present in the crocodile? So, the answer to this question is four. So, students, actually, these these are some exceptions. So, the heart of the crocodiles and the alligators, they have an exception, and they have got a four chambered heart which is very comparable to mammals and to the humans. So all the reptiles, they have got a 3.5 chambered heart, but the crocodiles and alligators, they have got a four chambered heart. Now students, in this particular slide I will be talking about this typical crocodilian four-chambered heart. Now, I'll be taking some of the physiological aspects of this particular heart also. Now, students, in this picture, you can very well see a crocodile is a very huge kind of animal, and this is a dissected crocodile. And here you can see all the viscera which is present in the crocodile, and here we have the heart. So if we see the structure, or you can see the typical structure of the heart of a crocodile, so you can very well see we have got the four chambers. Now this picture can show you the typical four chambers of the crocodilian heart. We have got a left atrium, we have got a right atrium, and we have got the left ventricle and the right ventricle. Now, why I have given you two diagrams of the crocodilian heart? Now, there is a difference. There is a difference in the physiology in these two conditions in which the hearts are present. Now, the first diagram is showing you the crocodilian heart in the normal state. And the second diagram is showing you this is the crocodilian heart in the diving state. When the crocodile is in the dive mode and it is underwater, and these crocodiles, they can dive for longer times. They can stay in water for longer times. And when they are staying in water, underwater, the physiology of the heart changes to some other condition which is maintaining an equilibrium of the blood flow to the brain in the crocodiles. So I have to give you some idea about the physiology of this particular heart. Now students, if we see it carefully, we are taking the very first diagram into account, which is the normal state of the crocodiles. Now here you can very well see, first of all, that the sinus venosus has a connection only with the right auricle. So there is no connection of the sinus venosus with the left auricle, and it is receiving from the body and the head, and this is a deoxygenated blood. So first, the deoxygenated blood is received by the sinus venosus, then it's go to the right auricle, it is going for the right ventricle. So there can be little mixing over here, as this is a partial septum in the case of reptiles, but in case of crocodile we have got a true septum over here. So there is no mixing in this particular case. So the stream is kept separate and the blood reaches into the right ventricle. Now from the right ventricle it is going into the pulmonary arch. So it can go to the lungs. This is the left lung. This will be going to the right lung. So this is very clear thing in this particular heart. But in the case of the left auricle, now here you can see the oxygenated blood from the lungs. It is coming into the left auricle, going to left ventricle. Then this is the foramen of Panizza. So this is the most important thing. So foramen of Panizza is like it is open. Now what happens that the blood flows into two arches now. One is going into this arch and the blood is flowing into this arch also. So the two arches, they are the right systemic and the left systemic arch. So it will flow into both the arches in the case of normal state. But here in the diving state, you can very well see the left systemic arch, it is having the deoxygenated blood from this side. And in the normal state, it is having the oxygenated blood. So this is the difference. Now, why is this difference present in the crocodilian heart? Because over here, the foramen of Panizza, it is closed. So what will happen? The oxygenated blood from the left side of the heart, it will go only and only into the right systemic arch, and it will go to the head parts also. So why is it so? Because in the diving state, the crocodile is in the need of more oxygen. While in the diving state, we have low of oxygen levels. So that is called as hypoxic conditions. So if the crocodile is in the hypoxia condition, we need more of the oxygen for the brain. So what in this particular situation has occurred that we have cut the oxygen levels or the oxygen transport to the left systemic artery, which is going for the stomach and the intestine. So while diving, we are not going for the, you can say, digestion process. So no oxygen is being wasted for uh, like stomach and intestines. So they are pumped the deoxygenated blood, and more of the oxygen through the left side is given to the brain. So that may increase the time of the diving for the crocodile. So this condition is for maintaining a proper, you can say, level of oxygens to the brain while the crocodile is in the diving mode. So this is the function of the foramen of Panizza. So this is a very important thing in case of the crocodiles. So that was the case of the normal state. So both the systemic arches are given the oxygenated blood, and the stomach and the intestines in normal state, they are getting the oxygenated blood. But if we talk about the diving situation, so here the crocodile is not eating or not digesting anything. So what is happening that left systemic arch or the artery is not getting any kind of oxygen. Now, why it is not getting that in diving condition, hypoxia condition, it maintains in the water and less of the oxygen is required by the body tissues. But here we need more of the oxygen for the brain. So this is the function of the foramen of Panizza. Now these are some of the variations in the reptiles, but I think that you must go for a typical crocodilian heart or a heart of a turtle or a 3.5 chambered heart in case of reptiles. So you can speak something about the exception in the crocodiles and alligators, but you must go for a 3.5 chambered heart in case of reptiles, as we have seen in the previous slide, which was red in color. Now, students, that was, I think, all about the reptiles. Now, in the next part of the video, I'll be talking about the birds and the mammals. Now, before going into the heart of the birds and the mammals, I must give you a concept that the hearts of both these groups, they are quite similar. There is a difference in the left and the right systemic. Otherwise, they are same in the structure. So we are starting with the heart of the birds and the mammals right now, as both are four-chambered hearts. So before starting the heart of the mammals, so there is a comparison of all the three types of hearts that we have previously discussed. The first is of the turtles, snakes, and lizards, and they are typically having 3.5 chambered heart. So here you can very well see these are the two atria, and we have got a partial septum which is present inside the ventricle. So they are typically 3.5 chambered. If I go for the crocodiles, they have got a four chambered heart as we have seen previously. And if I go for the mammals, they are again having a typical four chambered heart, which is not having any kind of you can say crossing over or any kind of physiological adaptation in this part, as we have seen in the crocodiles. So there was a foramen which was present in between the right systemic and the left systemic, but here in this particular case we don't have any kind of modification, or you can say complexity, in the heart of the mammals and the birds. Now students, if we go for the anatomy of a typical mammalian heart, you can very well see this is the cut-off heart of a mammal, and here you can see the right auricle. So this is the right atrium, and you may see this is the coronary sinus opening. So this is the sinus in which the whole of the deoxygenated blood from the coronary veins, it is being collected over here. So this is a part of the coronary system which is giving blood to the muscles of the heart itself. Now this is a tricuspid valve which is cut open into this particular right atrium. So this is present in the right atria, and we have got the right ventricle over here, and in this particular ventricle you may see a particular structure which is called as the trabeculae carne. So these are thick structures which are present in the ventricle. So here you can see the trabeculae carne along with the chordae tendineae. So I'll be giving you one more picture of the chordae tendineae. So this picture is showing you the particular thread-like structures which are present in the ventricle. These are the chordae tendineae of the heart, and these are quite profuse in the case of the mammals as compared to the reptilian heart. Now students, this particular diagram is a very simple, you can say, presentation of the circulation of the blood inside the heart of a mammal. Now if we go for the circulation inside this particular heart, so first of all, the deoxygenated blood from the body, it is going into the inferior and the superior vena cava. Now it is reaching from these vena cava into the right atrium. As we have seen in the previous cases also, the right auricle is always containing the deoxygenated blood. So here also we have got the deoxygenated blood in the right atrium. Now the blood is reaching the right atrium. From the right auricle, it is going through the tricuspid valve, as we have seen in the previous diagram. So through the tricuspid valve, it is reaching the right ventricle. Now here we have the semilunar valves. It is called as the pulmonary valve also. As this aorta, it is going for the lungs. So from the right ventricle, the blood, it goes into the pulmonary valve, and through this valve, it is reaching the pulmonary artery. Now you must have heard of this particular fact, that this is the only artery which is having the deoxygenated blood. This is an artery, but it is containing deoxygenated blood. So the blood will go to the lungs. Now here the oxygenation will occur in the site of the lungs. So from the pulmonary artery, it is going to the lungs for oxygenation, and after oxygenation, the blood will flow into the pulmonary vein. Now students, this vein is coming from the lungs to the left atrium. Now this pulmonary vein is a vein which is containing the oxygenated blood. So this is a, you can say, vice versa condition of a typical artery or a typical vein. So from this vein, the blood, it reaches the left atrium by these two veins. So these are two veins in number, and two are coming from this side. So they are opening into the left atrium. Now from the left atrium, we have got a mitral valve in between the left atrium and the left ventricle. So it is present over here. It is also called as the bicuspid valve. As we have a tri on this side, we have a bicuspid valve on this side. So this is also called as a mitral valve. And through this valve, the blood is reaching the left ventricle. So this is the oxygenated blood. So I am taking red circles for showing oxygenated blood. So after reaching the left ventricle, there is a big difference in the wall of the right and the left ventricles. So the wall of the left ventricle is quite thick as compared to the right ventricle. So you have to draw a thicker wall in case of the left ventricle as compared to the right ventricle. Now why is it thick? Because it needs more pressure to exert this blood to reach or to transport this blood to the whole body of the animal. So it should be more muscular. So from the left ventricle, it goes into this semilunar valve, and it is going into the aorta, which is leaving for the body and the head part. So this is the course of circulation in case of birds and mammals. Now students, ultimately it is the oxygenated blood which is going into the body and the head parts. So if I talk something about the pacemaker system of the hearts of the mammals, so it is the SA node, then we have the AV node, then we have got the bundle of His, then we have got the Purkinje fibers. As we have seen in the previous classes, we have discussed about this particular system from +1 and +2. Then in BSE also we have discussed this particular part. So I will not be going much into the pacemaking system, as we are discussing the evolution of the heart itself. So we are not going for the evolution of the pacemaker systems in this particular lecture. So after this pacemaking system, what we have is that if we recall the evolution of the heart, if we go for the amphioxus, so we have got many and many bulbuli, which are pumping the blood from the ventral aorta to the dorsal aorta. And in the due course of the movement of the blood, it gets oxygenated in the pharyngeal region of the emphysis. Now, next we have talked about the lamprey that was having a typical two-chambered heart with two accessory chambers. If we go further, we have got the fishes, we have got a typical two-chambered heart again. And if we talk about the amphibians, they had a typical three-chambered heart with some of the septal, you can say, modifications. Then if we talk about the frog or you talk about the lizard, it is having the three-chambered heart. Then if we go for the crocodiles, they have got a typical four-chambered heart. And if we talk about the chicken or the mouse or the humans, these are having the typical four-chambered hearts. And now here is a particular condition which is called as a blue baby. So this particular picture, it is showing you a defect which is called as the ventricular septal defect. So it is having a four-chambered heart, but in the case of blue babies, what has happened that there is a hole in the intraventricular septum. So what is happening over here is that in the case of ventricles of this heart, if you have a, suppose you have a, this is the right ventricle, this is the left ventricle, and in the left ventricle we have the oxygenated blood. In the case of right ventricle we have the deoxygenated blood, but in this septum we have a hole and the blood can be mixed from that particular hole. So this mixing, it is causing the coloration of the general coloration of the skin to some kind of a bluish tone of these babies. So that's why we call them as blue babies, as they have a hole in the intraventricular septum. So that can be cured by surgery in a particular age. So here we have taken a VSD condition, but in this particular case also we got a typical four-chambered heart. So here we consider it as we can say it as a 3.5 chambered, as it is having a partial intraventricular septum, but after surgery it can be a typical four-chambered heart. So students, that was all about the evolution of the heart among vertebrates, and I hope that you will go through this particular lecture in a very good manner, and if you get a question in your examinations, you may find it well to reproduce on the paper at that particular time. Thank you.